Hey everybody, welcome to the review video for week 8, the citric acid cycle. I'm recording this on early Sunday afternoon, trying to get this ready to be posted for Monday morning. So far the lecture watching's been trickling in, and I've really been seeing a mix of two things. One is students saying no questions at all, which can sometimes be a bad sign. Sometimes that means that it was just too much content, overwhelming, which is certainly understandable for this week's material. And the other comments were saying just that, outright, saying a lot of information, no specific questions, but a lot of information. So it struck me that perhaps it made sense to mimic what we did last week for glycolysis, but do it for the citric acid cycle, and just talk about the individual reactions, condense it down, distill it down, get away from all of the granular details, which are important in the first pass, but certainly not so important for the review, and just focus on the big picture of the citric acid cycle. So that's what we're going to try to do here. Here, what you're looking at is the citric acid cycle in its entirety, including what the pyruvate dehydrogenase complex is responsible for there at the top. And we'll come back to this at the very end. But let's take the citric acid cycle from the very beginning, and we'll use the same color schematics that we used before. So any slide that is rimmed or bordered in blue is going to be a control point. We won't talk too much about the control point here. We have a challenge question built around that later on this week that will allow us to focus on the control points and the regulation of the citric acid cycle a little more closely, but at least we'll hit on those steps that are regulated. And we'll go through the citric acid cycle step by step and talk about what's going on in each step to give it that big picture clarity that seemed to work well for glycolysis. So the first step of the citric acid cycle is not a step of the citric acid cycle. We end glycolysis with pyruvate, and the only thing that can enter the citric acid cycle is acetyl-CoA. So we actually have to bridge that gap. We have to go from pyruvate to acetyl-CoA, and that's what the pyruvate dehydrogenase complex is responsible for doing. So the PDC takes pyruvate and converts it into acetyl-CoA, and that acetyl-CoA then enters the citric acid cycle. So what you're looking at here is not formally or technically a step of the citric acid cycle, but is required in glucose metabolism to link glycolysis to the citric acid cycle. So briefly here in summary, this is a 4 or 5, depending on how you count it, 4 or 5 step process just to convert pyruvate into acetyl-CoA. The first step is a decarboxylation, so we lose this carbon dioxide off of pyruvate. It just leaves as CO2. And what we're left with is called hydroxyethyl. That's this portion of pyruvate here. And that hydroxyethyl is linked to the enzymatic cofactor TPP, yielding us hydroxyethyl TPP. Step 2 is a simultaneous transfer of hydroxyethyl to lipoic acid and the oxidation of that hydroxyethyl by lipoic acid. So here's lipoic acid, or lipamide. You can call it either. And that hydroxyethyl is transferred to lipamide. That recycles or regenerates free TPP. And once hydroxyethyl is on lipamide, lipamide oxidizes hydroxyethyl, and that results in acetyl. So now we have acetyl linked to the reduced form of lipamide. Two things are going to happen here. CoA is going to come in and take that acetyl. So it's another transfer. In other words, we transfer from TPP to lipoic acid to CoA. And since we had acetyl here, that acetyl, when it's transferred to CoA, gives us acetyl-CoA. That's what we were after. But what's left after that transfer is reduced lipoic acid, which needs to be regenerated and recycled back to its oxidized form, so the PDC can do this again. So in order to oxidize lipoic acid, FAD comes in and takes the electrons and protons from lipoic acid, oxidizing it. So now we can recycle that lipamide. This leads FAD in its reduced form. And then FAD is going to transfer those electrons and protons to NAD, which is our mobile electron carrier, and that's what we see here. And now the cycle is now the reaction is complete. So again, in summary, pyruvate, which is a 3-carbon molecule, loses a carbon dioxide, making what's left hydroxyethyl, and that hydroxyethyl is linked to TPP. Hydroxyethyl is transferred from TPP to lipoic acid, and lipoic acid also oxidizes hydroxyethyl, leaving acetyl. And lipoic acid itself is reduced. That acetyl group is transferred from acetyl-lipoic acid to acetyl-CoA, and lipoic acid is recycled back to its oxidized form by reducing FAD to FADH2. And FADH2 then transfers those electrons and protons to NAD, And so the net outcome of the PDC is 1 molecule of carbon dioxide leaving as waste, 1 reduced molecule of NADH plus H carrying electrons and protons to the electron transport chain, and acetyl-CoA. And that's the acetyl-CoA that we feed into the citric acid cycle. So now we're ready to begin the citric acid cycle. The first enzyme of the first step of the citric acid cycle is citrate synthase, and it is involved in a condensation reaction. It takes acetyl from CoA, so that recycles CoA alone. CoA leaves to do this again. And it takes the acetyl group, the enzyme does, and links it to oxaloacetate. Oxaloacetate is a 4-carbon molecule, acetyl is a 2-carbon molecule, and this yields 6-carbon citrate. So acetyl is transferred onto oxaloacetate, that gives us citrate, and the CoA group recycles back to the PDC. Step 2, that citrate is isomerized. So a water molecule is removed and then added, but it's added in a different way than it was before. So net result, no atoms gained, no atoms lost. Citrate becomes isocitrate. Oh, and I should say, we're rimmed in blue here, so the PDC is a regulated step, and step 1 of the citric acid cycle is regulated as well. Step 2 is not a regulated step. It's in equilibrium. So the net result of step 2 of the citric acid cycle is isocitrate, an isomerized form of citrate. In step 3, a regulated step, isocitrate is going to be oxidized. So let me say this specifically here. We had some confusion last week. Dehydrogenase enzymes, such as isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, even the dehydrogenase that we encountered in glycolysis in step 6. The PDC stands for pyruvate dehydrogenase complex. Those are not dehydration reactions. Dehydrogenase is meant to say that a dehydrogenation reaction is occurring. In other words, we're stripping protons from these substrates. And the reason why we're stripping protons is because where the protons go, the electrons follow, or vice versa. So dehydrogenase enzymes actually catalyze redox reactions. Whenever you see dehydrogenase, think redox. And such is the case here. So step 3 of the citric acid cycle is catalyzed by isocitrate dehydrogenase, and this is our first oxidative decarboxylation. We're losing one of those 6 carbons, so we go from 6-carbon isocitrate to 5-carbon alpha-ketoglutarate. And we're losing a carbon dioxide. That's how we come out to a net even number of carbons. And we're also reducing our mobile electron carrier NAD plus to NADH plus H. So step 3 of the citric acid cycle, isocitrate is oxidatively decarboxylated. We go from 6 carbons to 5. We lose 1 carbon as carbon dioxide, and we're reducing NADH plus H. We're oxidizing alpha-ketoglutarate. Step 4, also regulated. Yet another oxidative decarboxylation. Alpha-ketoglutarate is now oxidatively decarboxylated to succinyl-CoA. This is catalyzed by the alpha-ketoglutarate dehydrogenase complex, which works almost identically to the PDC. We're going to talk about this in much more depth in our challenge questions this week. So this is another oxidative decarboxylation. We lose another carbon dioxide, and we oxidize alpha-ketoglutarate, meaning we reduce NAD to NADH plus H. CoA is also a cofactor here. It's swooping in and accepting succinyl in a high-energy bond. So there's a lot of parallels between what the PDC does to make acetyl-CoA and what the alpha-ketoglutarate dehydrogenase complex does to make succinyl-CoA. Again, we'll talk about that later on this week in the challenge questions. It's also worth pointing out that from a carbon perspective, glucose is now gone. A single molecule of glucose entered glycolysis. It was essentially broken in half and turned into 2 molecules of 3-carbon pyruvate. So we didn't lose any carbons there. 6-carbon glucose became 2 pyruvates, 3 carbons each. When pyruvate became acetyl-CoA, we lost carbon dioxide. We lost 1 carbon each. So we went from 6 total carbons to 4, because remember, we made 2 pyruvates for every 1 glucose, and those 2 pyruvates are going into the citric acid cycle. So everything we do, pretty much everything we do after step 5 of glycolysis is done in parallel. So when we lose a carbon dioxide from pyruvate, we're losing 2 carbon dioxides from 2 pyruvates. We're left with 4 carbons from that original glucose. We lost 2 more carbons in the parallel reactions of step 3, and we lost the final 2 carbons of glucose in the parallel reactions of step 4. So the carbons of glucose are now gone. We've converted, or I should say stripped, the glucose of electrons, and will continue to do so. So step 5 of the citric acid cycle, succinyl-CoA is broken and yields succinate. So CoA leaves as a leaving group. And that's a high energy bond between CoA and succinyl, and that energy is captured in GTP synthesis, and that GTP is immediately used to phosphorylate ADP to ATP. This is our 1 ATP net gain of the citric acid cycle. Not what the citric acid cycle is about, but with so much energy being released there in that CoA bond, we capture it as a single molecule of ATP indirectly. Step 6 now, I believe. I've lost count myself. Step 1, Step 2, Step 3, Step 4, Step 5, Step 6. Yep. Step 6 is the oxidation of succinate to fumarate. This is catalyzed by succinate dehydrogenase, and here, and only here, our electron carrier is FADH2. Well, FAD plus is reduced to FADH2. FAD is not a mobile electron carrier, so this pair of electrons and protons is stuck on succinate dehydrogenase, and that's going to become important after exam 2. When we talk about the electron transport chain, we'll actually see that succinate dehydrogenase plays a role directly in the ETC as well. But we're left with fumarate. Fumarate is then going to be hydrated to become malate, so we add a single water molecule to fumarate to become malate. That's catalyzed by fumarase. And finally, the last step of the citric acid cycle is the regeneration of oxaloacetate through a redox reaction. So malate dehydrogenase, there it is again, right? Dehydrogenase, we're stripping protons, we're stripping electrons, we're doing a redox. Malate is oxidized to oxaloacetate, and NAD, once again, will accept those electrons and protons and become the reduced form NADH plus H, and ultimately take those electrons and protons to the electron transport chain. And we've regenerated oxaloacetate. So we've completed the cycle. So as busy as this might look, that's pretty much it in a nutshell. We just discussed it. We've got the PDC, arguably the most complex single step of this material, because a lot is going on as we convert pyruvate into acetyl-CoA. But the net result of all of that is we are stripping a carbon dioxide or a carbon off of pyruvate, going from 3 carbons to 2. And we're taking what's left, acetyl, and linking it to acetyl-CoA. We're getting carbon dioxide as a waste product, and we're getting reduced NADH plus H as a byproduct, as a valuable byproduct of that reaction. We take that acetyl, we separate it from acetyl-CoA, and we link it to oxaloacetate. So here's oxaloacetate coming into the reaction. Here's the acetyl group of acetyl-CoA. CoA is leaving as a leaving group, and we need a water to do this as well. And we make citrate. So citrate is yielded. We go from a 4-carbon oxaloacetate to a 6-carbon citrate. That citrate's going to be isomerized in step 2. And we go from citrate to isocitrate, isomerized citrate. Isocitrate, I've lost my pointer. Isocitrate is going to be oxidatively decarboxylated and go from 6 carbons to 5. We lose carbon dioxide there as a leaving group, and we yield a reduced form of NADH plus H. This is our first oxidative decarboxylation of the citric acid cycle. Then we do it again in step 4. We strip another carbon dioxide off of alpha-ketoglutarate. We reduce another set of NAD plus to NADH plus H, and that yields this high-energy succinyl-CoA. Step 5, we break that CoA bond and capture that energy as GTP. We'll ultimately use that GTP to make a single ATP. CoA leaves as a leaving group, and we're left with succinate. Succinyl-CoA cracks to succinate. From this point on, we're just regenerating oxaloacetate in the next 3 steps. Succinate is going to be oxidized to fumarate. We're going to steal those electrons and protons and keep them on succinate dehydrogenase. Fumarate is going to be hydrated into malate. Malate is going to be oxidized to oxaloacetate, and we come back to the cycle here. So what do we get out of all of this? Well, we actually get out of this 10 electrons and protons. That's what we're after in the citric acid cycle. 1 pair of electrons and protons is here in the PDC. The second pair, getting us to a total of 4, is the first oxidative decarboxylation, a total of 6 after the second oxidative decarboxylation. A total of 8 at succinate dehydrogenase, and our 10 total electrons and protons that we've stolen from glucose in the last step of the citric acid cycle, step 8, as we regenerate oxaloacetate. And we also make that 1 GTP, which is converted into ATP. We lose a total of 3 carbons, 1 carbon dioxide left by the PDC and the other 2 carbon dioxides in these oxidative decarboxylations. Remember, we're doing 2 citric acid cycles for every 1 molecule of glucose because of that dehap to gap isomerization in step 6 of step 5 of the citric acid cycle. And so that's going to have this going in parallel for us. So when we are losing 3 carbon dioxides in the citric acid cycle, we're actually losing a total of all 6 carbons of glucose because we're doing this with 2 pyruvates for every 1 molecule of glucose consumed. And the regulation steps are the PDC step here. We're regulating the pyruvate dehydrogenase complex. We regulate citrate synthase, and we regulate the 2 oxidative decarboxylations. So 4 points of regulation here in the citric acid cycle. And in a nutshell, all of these regulation schemes are responding to cellular energy levels. They're looking at reduced NAD levels and ATP levels. If ATP levels are high, that downregulates the citric acid cycle. If ADP levels are high, that upregulates the citric acid cycle. If NAD plus levels are high, that upregulates the citric acid cycle. If NADH plus H is high, it downregulates. But we'll talk about that in the context of the challenge question. So we'll come back and review the regulation of the citric acid cycle in much more detail in the challenge questions. But that's pretty much what we're after. Now, there's far more important stuff that you're responsible for in this lecture. We talk about regenerating oxaloacetate from pyruvate, right? We can build more citric acid cycles by converting pyruvate directly into oxaloacetate instead of going into acetyl-CoA. We can get acetyl-CoA from the oxidation of fatty acids and the breakdown of proteins. We talked about how the citric acid cycle is a hub for catabolism and anabolism, how we can make amino acids from different precursors of the citric acid cycle. So there's a lot more to this material that we definitely want you to have full command of. But it made sense, I thought, to keep the context and perspective of glucose metabolism at the forefront for the review and continue to build this story of glucose metabolism. Glucose to glycolysis, to the PDC, to the citric acid cycle, and then after the exam, we're going to see to the electron transport chain, to oxidative phosphorylation, and that's where we're going to make a lot of ATP. So please don't leave this video thinking that this is all you need to know about the citric acid cycle. It's better to say that this provides you with the context of the citric acid cycle, and then you can fill in the more relevant, more pertinent details that you need to know in this context. So let's make sure the citric acid cycle itself is clear first, and then you can go back and review those finer points and finer details as to where the citric acid cycle fits as the hub of all cellular metabolism in eukaryotes. So you know what to do at this point. Last review session of Unit 2. If you have any questions about this material specifically, or any questions about these 2 lectures more in general, feel free to let me know in the dialog box that's about to pop up. And if not, the challenge questions are going to give us plenty of opportunity to continue to practice with these concepts. And then you'll hit the books hard next week as you prepare for the second unit exam. And that'll lead us into the last 3 weeks of instruction of the course and take us all the way to the summer. So as always, thanks for watching, and I'll see you all on the flip side for the challenge questions.